Hey everybody, we are going to talk about different types of mutations. Now, we already discussed a few terms. Let me just briefly go over those. Directed mutations. Well, let me rephrase that. Mutations can be directed, they can be random, they can be induced or spontaneous. Now, we use these terms when we were talking about the nature of mutation. So, do they occur, say, randomly throughout the genome, or do they occur in specific genes in response to a certain environmental agent? In that case, they would be directed to certain genes. And we saw that because of the Lederberg experiment, or the experiment performed by the Lederbergs, is that, mutations are thought to occur mostly randomly. And we did discuss there are some situations where they are directed, such as somatic hypermutation, but for the most part, they occur randomly. So these two terms we're using to address this concept of the nature of mutation. So these things right here, these are induced and spontaneous. So how mutations form? So do they occur spontaneously in the cell, or are they induced by external agents? And both of these can occur. So cells, just being normal cells, can make mistakes and things can happen, and DNA can be damaged, or the polymerase can make a mistake, and we consider those spontaneous mutations. So any mutation that is not an induced mutation is a spontaneous mutation. Induced mutations, those are caused by some external agent, some agent that is external to the cell, a chemical that's in the environment, or a physical source such as radiation, ionizing radiation, or even UV light. Now, in this video, we are going to talk about types of mutations, so ways to classify mutations. We have all these terms that we want to discuss right here, and this is problem, in this spring 2020 version of the course. This problem is lecture 16.B. And okay, we need to know what these mutations or what these terms mean. And why do we care? Well, if you know what these mean, then you'll be familiar with the different effects mutations can have on cells and organisms. So before we start going through that list of mutations, it's important to have just an idea of what we are going to consider a mutation to be. So let's take a wild type sequence, and we will consider the wild type sequence of a gene, say, to be the one that is the most common in a population. So that will be the wild type sequence. And we will consider a mutant sequence to be anything that is not wild type. So mutant sequence is any sequence that is not the wild type sequence. And there are some caveats there, right? So if 90% of the population, let's say, has a G at one position and 10% has an A at the other position, is this a mutation or is it a polymorphism? So we don't really want to get into those minor details of what is a mutation and what is just a natural polymorphism in a population. We are going to consider anything that is not the wild type sequence. We will consider that a mutant sequence. And as we classify these different types of mutations, we will see that not all mutations have an effect on fitness of the organism. Some are neutral. So just because there's a mutation doesn't necessarily mean that is bad for the organism. And some mutations can be beneficial. So let's start talking about, or going over, these different types of mutations. So first, let's take gain of function mutation. And I forget exactly why, but we did cover this briefly in the beginning of the course. So a gain of function mutation. I think we were talking about how something could be dominant, how a mutation could be dominant over the wild type sequence. But here, let's review this again, a gain of function mutation. Well, one example, and there could be a number of different ways this could happen, is let's say we have a protein here, and this is a protein, and there's an amino acid in this protein that is modified. Let's say it's phosphorylated. There's a phosphate group put on that amino acid at that position. And let's say when that's phosphorylated, the protein is locked in a functional conformation. And okay, now let's say there's an enzyme in the cell that finds this, when this other enzyme is expressed, and it takes that phosphate off, a dephosphorylase enzyme takes this off. And when that phosphate is off, let's say this protein adopts a new conformation. And this is the non-functional conformation. So when that amino acid, whatever it is, when it's not phosphorylated, the non-functional conformation is adopted. Conformation is the structure of the protein. So when that amino acid is not phosphorylated, it's in the non-functional conformation. So this is very common for proteins to exist in two forms, functional and non-functional. And the shape changes based on, which amino acids are modified in certain ways. And one conformation is non-functional, another is functional. So if we had a mutation, let's say that locked this protein in the functional form, so it couldn't go back and forth, it was always in this functional conformation. So a mutation in a codon changed the sequence of the amino acid, changed one of the amino acids, so it couldn't go back and forth. It's locked in the functional conformation. That would be an example of a gain of function mutation. Now you can imagine the opposite, loss of function. So a loss of function mutation would be any mutation, say in a gene, that locks that protein, changes an amino acid, so it's locked in the non-functional conformation. It can never function. So that would be an example of a loss of function mutation, a mutation in an amino acid that locks the protein into a non-functional conformation. Now there are different grades, right? Loss of function mutation. Maybe it's a mutation that just inhibits the functional conformation from forming, but it can form sometimes, and it just sort of decreases the ability to move from non-functional to functional. Well, that would also be loss of function, but a null mutation would be a mutation that completely eliminates function of the protein. So we can have different grades of gain of function and loss of function. But a loss of function mutation that completely results in complete loss of the function of the protein, that's a null mutation. And we assumed that when we were talking about Mendel in the first part of the course, that most of Mendel's mutations were null mutations. I don't know that for sure, but we assume that. And it will be interesting to look in the future if people have uncovered or discovered all of Mendel's mutations, the molecular explanations for all the different phenotypes in his pea plants. Okay, so enough of the aside on Mendel. Now let's talk about point mutations. So when we say something is a point mutation, well, essentially what we are saying is we are changing one base in the sequence. So let's say this is the wild type sequence, just a bunch of A's there. Now, if we change one of these A's to any other base, that would be a point mutation. So I'm going to change that one. I will change it to a C. Okay. So we have a point mutation here. This is the wild type sequence, this is the mutant sequence, there's just one base changed. There's a point mutation at this position. Now, point mutations can be classified in certain ways. So a transition mutation is when a purine is changed. Why am I having trouble spelling changed? Is changed to a different purine. So a G is changed to an A, or an A is changed to a G. Similarly, a transition mutation is also when a pyrimidine is changed to a different pyrimidine. So those are both types of transition mutations. Transversion mutation, you can probably guess what that is, right? So that's going to be when a purine changes or mutates to a pyrimidine, or vice versa, a pyrimidine changing to a purine. That's an example of a transversion mutation. Now, we have insertion and deletion mutations. Right, so those aren't point mutations. We're not changing specific bases from one base to another. Insertion mutations are we're inserting bases or nucleotides into a sequence, inserting nucleotides that weren't there. In deletion, we're deleting nucleotides that were there. And these can cause what we call frameshift mutations. If these occur in the coding region of a gene, they can cause a frameshift mutation. So frameshift mutations are specific for coding regions of genes. Now, insertion and deletion mutations they can occur anywhere in the genome. They don't need to be in a gene. They can be between genes. They can be in regions of the genome that don't have many genes at all, like the centromeric regions or telomeric regions. But when they occur within the coding region of a gene, they may cause a frameshift mutation. And whether or not they cause a frameshift mutation depends on if insertion or deletion occurs in a multiple of 3. Now let me explain more about that. So let's take a closer look at frameshift mutations. So again, discussing frameshift mutations here. So if this is the sequence of a gene, let me put some codons in here. So we're looking at the DNA, we're not looking at the RNA sequence. And I'm not going to diagram I'm not going to put the bases in for the complementary strand here. I just want to focus on this coding region of this very small coding region, this hypothetical coding region, theoretical coding region here. Here's the start codon. Again, we're looking at DNA, so we're seeing T's instead of U's. Let's say this is the stop codon here. And we have 1, 2, 3, 4, 5, 6 different codons in this coding region right here. Now let's assume this is the wild type sequence. And if we insert one base, just one base, let's say at this position right here. Let's say we insert a T there. See what happens. I'm going to put the T in right here between this ATG and this CGA. And then I'm just going to copy these bases down right here. Okay. So everything else, everything about the sequence is identical between these two, except there's a T here where there was no T before. Now, as a result, okay, we have the start codon here. So translation, if this was transcribed into an RNA molecule, T's changed to U's, and this messenger RNA was, and the resulting messenger RNA was translated, okay, this would be the start codon. And then this is the second codon, right? Because the ribosome reads the codons bases 3 at a time. So this is the second codon, this is the third, this is the fourth, fifth, sixth codon, and so on. So you can see we've shifted the reading frame. So these codons, the first codon, the start codon is the same, but the second codon is different, the third codon is different, fourth is different. So we have significantly changed the coding sequence of this gene, and the protein that results is going to be very different. It's going to have very different amino acids in each position. By chance, some of the codons may line up and be the same, but most of them are going to be different. And we may even get a stop codon, an early stop codon in here. Well, this is a very small protein being produced, right? So we're not likely to see a stop codon, but proteins can be very long, very long. And when you have a frame shift, well, then you can get an early stop codon caught very early in the protein. Okay, so we insert one base, we get a frame shift. Now, what if we inserted 3 bases? I bet a lot of you, if you're watching the video and instead of just learning from the notes directly, can probably guess what's going to happen if we should insert, say, 3 bases right here. So let's put 3 T's in instead of 1 T. And then I put 3 T's in after this G, and I'm just going to copy the rest. Well, I hope I hit record on this. Let's see. Yes, I did. Okay, now we inserted 3 here. First codon is the same. Second codon is different. But let's see what happens now. This second codon right here is now at the 3rd position. This 3rd codon is at the 4th position. So for the most part, you know, we didn't shift the reading frame. We just inserted a completely different codon between the first and the second codon. So first and second codon here, by inserting 3 bases, we keep the reading frame the same. All the non-inserted codons are identical to what's in the wild type sequence. We just have these extra 3 bases. So whenever we insert bases in a multiple of 3 or delete bases in a multiple of 3, we don't shift the reading frame. The reading frame stays the same, and most of the amino acids in the protein will stay the same as long as we're inserting in a multiple of 3. And that is due to the nature due to the nature at which ribosomes translate a messenger RNA, right? They read 3 bases at a time. So if we insert 3 bases, then we don't change the reading frame. Now there is a problem in, let's see, problem 1532, which is going to take a look at frameshift mutations in more detail. So a frameshift mutation occurs in the very beginning of a gene, a coding sequence, instead of the end. You know, a frameshift mutation that occurs in the beginning of a sequence is more likely to have a detrimental effect on the function of the protein than is one that occurs at the end. And you could probably see why that's the case, right? So if you change all the codons at the beginning of the gene, or from the beginning of the coding sequence, well, all the codons after that point are going to be different. So if the frameshift is towards the end of the coding region, well, there aren't that many amino acids left in the protein for translation, and maybe the protein can get by without those few amino acids being identical to wild type. Again, 1532 is the problem, and there is a YouTube walkthrough video of that problem available, so you can learn more about frameshift mutation. So a frameshift mutation isn't necessarily a mutation that occurs in the coding region if it occurs in a multiple of 3. So it has to occur in the insertion or deletion has to occur in a non-multiple of 3 in order to be a frameshift mutation. It must change the reading frame to be a frameshift mutation. So I think that's it for frameshift mutations. Let's go on to, where is my list? Okay, so we have other types of mutations, ways to classify mutations in coding regions of genes. And these are fun because when you're talking about, if any of you take population genetics and how beneficial mutations or neutral mutations and all these things spread through populations, the coding regions of genes become important. So we're going to see a bunch of terms that you would see in courses such as population genetics in this part of the video. So coding regions of genes. Let's take a look at some other mutations other than frameshift mutations that can occur in the coding region of a gene. So here is a coding region. Again, I'm not going to diagram the complementary strand. Make sure I don't put in a stop codon until the end. Okay, there we go. So again, we're looking at the DNA molecule, not the RNA that's transcribed from this. Now again, here's the start codon right here, and here's the second codon. Let's just we could take a look at any of these codons if we wanted to talk about these terms I'm going to bring up next, but let's just look at this one right here. So CGA. So let's say there's a mutation that changes this A. So A to T. So a point mutation at this point right here. A to T. So what happens? We're going to call that a sense mutation. Now, we can also call it a synonymous. Two terms mean the same thing. Sense mutation and synonymous mutation. Why? Why can we call those? Why is this a sense mutation and a synonymous mutation? So CGA codes for arginine. So if you were to pull up a table of genetic code, you would see CGA codes for arginine. CGT also codes for arginine. So if a point mutation occurs in the coding region of a gene, changes a codon in a way that doesn't change the amino acid it specifies for, then that is a sense mutation and a synonymous mutation. You know, same terms, they mean the same thing. So those are pretty easy to understand, and they have to do, or they're a result of the genetic code being degenerate. I think some of you probably remember that from the lecture on the genetic code before spring break. So let's take a look at this one right here. What if this is a C to G mutation at this position? Well, CGA, like we already said, codes for arginine. So again, we're talking about this codon right here, so we'd be changing that C to a G. GGA codes for glycine. Again, you don't need to remember those, right? But if you looked at a genetic code, you would see that these are different, these codons code for different amino acids. Now, this type of point mutation would be considered a missense mutation. So a point mutation in the coding region of a gene that changes a codon in a way that the new codon codes for a different amino acid is called a missense mutation. We can also call it a non-synonymous mutation. So a missense mutation is a type of non-synonymous mutation. Now there's also another type of non-synonymous mutation, and that's called a nonsense mutation. So let's take this C right here, let's change that to a T. So a C to T mutation at this position. Well, we already said CGA codes for arginine. Now the mutated codon, TGA, that's a stop codon. So a point mutation in the coding region of a gene, a point mutation in a codon that changes the codon from coding for an amino acid to coding for a stop codon is a nonsense mutation. And the nonsense mutation is also a type of non-synonymous mutation. So two types of non-synonymous mutations, right? A nonsense mutation and a missense mutation. Missense is when one codon goes from coding for one amino acid to another. Nonsense is when a codon goes from coding for an amino acid to a stop codon. Now, I like those terms. I think they're kind of fun. Now, let's take a look at some others. These ones are easy. Let's take a region of the genome, and let's say in the wild type sequence, there's an A. Just at that spot right there. It doesn't matter where it is. It's in the centromere, it's in a gene, it's between genes, an intergenic region. Let's say there's an A there, and there's a mutation. Let's say a mutation occurs. Now there's a G there. And a C there. Okay, now if this mutation has no effect on the fitness of the organism, we call that a silent mutation. And you can have silent mutations in genes, in centromeres. They can be anywhere. And as long as, and if it doesn't cause, and the mutation doesn't cause an effect on the fitness of the organism, it's considered a silent mutation. Silent mutation, no effect on fitness. And, for example, what about a non-synonymous mutation? Non-synonymous, I feel like I might be going off track a bit, but let me just diverge a little bit. So a non-synonymous mutation, even in a mutation that changes a codon from coding for an amino acid to coding, let's say, for a different amino acid, that doesn't necessarily have an effect on fitness of the organism. We don't know. We don't always know. If we can't detect an effect on fitness, it's a chance, okay, it could be a silent mutation. Even though it's not synonymous, it may not have an effect on fitness. So silent mutations are any mutation that they could have they could change the amino acid. They could change the codon from coding for one amino acid to coding for another. If it has no effect on the fitness of the organism, it's a silent mutation. Another word for silent mutation is neutral mutation. So those terms are synonymous. So just two more things, very easy terms: somatic mutation versus germline. So somatic mutation is any mutation that occurs in a somatic cell. So what is a somatic cell? It's any cell that's not part of the germline. Somatic mutations, a muscle cell, I don't even know, other skin cell, a cell in your eye, any mutations in those cells are somatic mutations, and importantly, they are not passed on to your children, right? They're not passed on to the next generation. They're still bad, right? Somatic mutations can lead to cancer, but they're not passed to the next generation. Any mutation that occurs in the germline, in germline cells that are directly responsible for producing gametes, that give rise to gametes, now those mutations can be passed to the next generation. So I don't think, these terms are too difficult, but, by understanding them, they help us see all the different types of effects, or a nice introduction to the types of things or ways mutations can have influences on function of the cell, changes in codons and things like that. Okay, so in the next video, I think we're going to take a look at genetic complementation, and maybe one more video after that, just briefly on nucleotide excision repair. But I might be able to fit both those things into the same video. Okay, see you in the next video.